This is Module 6, Eukaryotic Gene Regulation. In this video, 6-3, Regulating Genes with DNA Binding Proteins, I'll be talking a little bit about the DNA sequences that are regulating open reading frames, and also about the proteins that bind to them to either increase or decrease gene expression. Eukaryotic gene expression can be complicated because of the two different types of promoters that exist upstream of each open reading frame and all of the different types of elements within those promoters that can control expression. And so recall from a previous module that we have a core promoter, I think this was in the transcription module, and also a regulatory promoter for eukaryotic genes. For every eukaryotic gene, each has its own core and regulatory promoter. Within the core promoter, there's more consensus sequences or sequences that are common to every promoter in the genome. Within the regulatory promoter, that's where you start to see diversity in the types of DNA elements that control expression. Just to remind you, the basal transcription apparatus, which is the TBP protein, will bind to the TATA box in the core promoter to initiate transcription. But it has to also bind with several other different transcription factors, and it has to associate with proteins bound to the enhancer region, which is much further upstream of the core promoter. And so I sort of touched upon this in the previous module about transcription, but let's talk about these proteins in a little bit more detail. Mediators are proteins that bind to the basal transcription apparatus shown here. And this helps the transcription apparatus, which contains the RNA polymerase, interact with other regulatory regions in the regulatory promoter and upstream. Some of those interactions occur with coactivators, which are connecting the mediator to what's happening at the regulatory promoter, and also other transcriptional activators that connect the mediator to what's happening at the enhancer region. And so transcriptional activators are proteins that bind to enhancer regions in the DNA. So enhancer refers to a piece of DNA, a sequence of DNA. Activator proteins then interact with mediator proteins that are bound to the transcriptional apparatus in order to stabilize this transcription apparatus. Co-activators are proteins that also bind to the mediator and other transcriptional activators at the regulatory promoter. But co-activators will basically attach the transcription apparatus to the regulatory promoter that's close by, and then the mediator helps to interact with the enhancer that's very, very far away. Okay, so let's take a closer look at the diversity that I keep talking about that exists within regulatory promoters of eukaryotic genes. There are consensus sequences within the core promoter that are common to all eukaryotic genes, such as the TATA box. That's where the TBP protein is binding to stabilize the basal transcription apparatus and initiate transcription. However, within the regulatory promoter, the types of DNA elements that can exist there can be very different depending on what the gene is that they are attached to. For example, here we have this regulatory promoter which helps to control the expression of thymidine kinase. So thymidine kinase will be so there's lots of different sites, consensus sites within the regulatory promoter. There's the OCT site, which binds to transcription factors that either increase or decrease expression. There's the GC site, which always binds activators, transcriptional activators, and the CAT site, which also binds transcriptional activators. And so you can understand and appreciate that this gene can be increased in gene expression in response to some signals by binding the GC and CAT region. It can also be decreased in response to some outside signals by binding repressors to this OCT region. Note how different that is from this gene here, which is the SV40 gene, and also how different that is from this histone gene. And so different types of consensus sequences within the regulatory promoter will influence gene expression. And that can be either increased gene expression or decreased gene expression depending on what's available. This one, the SV40 gene, can only have an increase in gene expression. You can't actually suppress gene expression of this one because there is no DNA site within it that will bind a transcription factor that reduces gene expression. So that's just one example of how things can be really different in the regulatory promoter regions. Let's take a look at an example of one of these types of regulation. So let's look at a gene. All right, so this is the gene that controls production of or metabolism of galactose, which is a sugar that can be oxidized to create ATP. We have a couple different regulators to keep track of here. So we have the UAS, which is the upstream activating sequence in DNA. This is probably also an enhancer region, this UAS. This is a sequence of DNA that when proteins bind to it will help to alter the rate of transcription. There's also GAL4, which is a transcriptional activator. When GAL4 is bound to the UAS or the enhancer region, this will increase gene expression of enzymes that help to break down galactose. GAL80, where is GAL? GAL80 is right here, and GAL80 will bind to GAL4. It can also bind to GAL3. It is a repressing protein, and so GAL80 will actually block the activation of transcription caused by GAL4. And then GAL3 is a protein that binds to galactose in order to increase gene expression of the proteins that metabolize galactose. So let's break this down and try to assign some terms and figure out how these things are working. So UAS, this is an enhancer. This is a sequence of DNA that when proteins are bound to it should result in an increase in gene expression. GAL4 is a transcriptional activator. So when it binds to the enhancer, it will increase gene expression. GAL80 is a transcriptional repressor, and so it reduces gene expression. And GAL3 is a transcriptional activator that only will increase gene expression in the presence of galactose, which makes sense. If you have galactose in the cell, you need enzymes to process it, and so galactose itself is also an activator of gene expression. And so in the absence of galactose, there's no galactose in the cell. So there's no reason to make these enzymes if there's no substrate to act on. And so in the absence of galactose, GAL80, the repressor, will block the activation of transcription coming from the enhancer region. And so when GAL80 is bound, you don't get any transcription of this gene that helps to process galactose. However, when galactose is now present, it will do one of two. This results in a couple of different things. Galactose will bind to GAL3, and GAL3 helps to, get this, repress the repressor. So when galactose is present, we want to make this enzyme so that we can process galactose. So when galactose is present, it will help the GAL3 protein repress GAL80. GAL80 falls off of GAL4, and now GAL4 is allowed to activate transcription. I recommend you write out the steps of what happens in the presence of galactose and what happens in the absence of galactose in order to keep this straight in your head. So let's talk about these repressors then. And again, the repressor in this example is GAL80. When a repressor protein is bound to sometimes repressors can bind other proteins, like in this example, it's binding to the activator proteins. Repressor proteins can also bind to DNA regions, called silencers. Silencers are basically act like the opposite of an enhancer. Silencer is a region of DNA that binds to a repressor protein. And repressor protein binds to silencer, transcription is blocked, and gene expression is reduced. And so basically the silencer-repressor interaction has the opposite effect as the enhancer-activator interaction. There's yet one more there's another region of DNA referred to as an insulator. And insulators can actually block the action of enhancers when the insulator lies between the enhancer and the promoter. So let's kind of break this down. Here we have a promoter and an enhancer. And so when activators are bound to the enhancer, it will increase transcription at the promoter in this direction. Here's another gene that's being transcribed in the opposite direction, gene B. It has a promoter and an enhancer also. And so when activators are bound to the enhancer, that increases expression at the promoter, or it increases binding at the promoter and therefore gene expression. When an insulator comes between the enhancer, so again, when the insulator lies between the enhancer and the promoter, it will block expression. And so this enhancer region has the ability to not just enhance this promoter, but also enhance this promoter over here. It doesn't really, it's not specific to this promoter. Enhancers can enhance any promoter. And so this insulator lies between enhancer 1 and this promoter for gene B over here. If there is an insulator binding protein bound to this insulator, then you can activate gene expression of gene A still through this enhancer action, but activation of expression from gene B is blocked because this enhancer cannot enhance this one. One last piece of DNA that I want to mention that helps to regulate gene expression are response elements. In eukaryotes, sometimes we need to activate several genes in order to respond to a single stimulus. For example, if you become ill with something, you need to be able to activate all of the genes that are responsible for increasing the inflammatory response. Let's say you come across a heavy metal and you need to activate all kinds of genes that help to detoxify the bloodstream of that heavy metal. Wouldn't it be nice if you could activate expression of all of the genes required to solve this problem all at once? And the answer is you can, as long as the same response element exists in the promoter of all of those genes. All you need to do is turn on one protein that will bind to all of those response elements, which will help to activate a whole set of genes as opposed to just one. So this means that genes that need to be expressed at the same time in eukaryotic cells are able to respond to the same stimulus because they all have the same code in their regulatory region. And so that same code will respond to the presence of one activating protein. So let's take a look at what this looks like. So here is a metallothionein gene. Metallothionein helps to detoxify the bloodstream from heavy metal poisoning. And so upstream we have the core promoter region, which has the title box. That's common to all genes. And then in the regulatory region of this metallothionein gene, we have a couple of enhancers. One is the TRE enhancer, which one type of transcription factor binds to TRE and stimulates transcription of this gene. And then we have, so this is a type of response element. And so any gene that has the TRE response element in it will respond to the presence of AP1, this activator protein. And so this metallothionein gene will respond to it. And then another type of detoxification gene will also have TRE in its promoter, and it will also respond at the same time. That's also how the GREs work. So these are glucocorticoid response elements, and they help to stimulate transcription by binding to steroid hormones, which increase transcription for some genes. These MREs are metal response elements. I'm sorry, yeah, these are metal response elements. So that's an unfortunate that matches this multiple response element, but this means metal response element. I'm sorry. Where activator proteins that are turned on in response to heavy metals will all bind to any promoter that has an MRE in it. And so this helps to coordinate gene expression in response to stimuli that require many gene products in order to control.